one thing I want to add to that kind of folds into this lecture is what's the difference between what you will normally see in your sperm searches that you've been doing for years and doing it on the LMD. And this is what's nice about Pat going through kind of the microscopy, because if you're kind of a little weak on the microscopy, this is a system that you really have to be brushed up on, because one, you're not doing it with a cover slip, and two, you're looking through a glass slide, and three, there's also a pen film that creates architecture. So there's some little hurdles that you'll have to overcome and get used to when looking at these particular slides and identifying your sperm. So this is a picture. This is Christmas tree stain. It doesn't come, of course, the color doesn't come out as well as on the screen here, of sperm and epithelial cells. And just briefly, we'll go over this a little more detail later, but why is cell separation important? DNA mixtures are frequently encountered in sexual assault evidence, and everyone knows that, and there's probably huge backlogs of it in most of your facilities. And the idea is that we want to identify individuals involved in the crime. Genotype mixture interpretation and statistical analysis can be challenging and time-consuming, especially when you have mixtures, if you have mixed genotypes. So the idea is separation of the different cell types can provide cell fractions from individuals involved in the crime. That's what we're here for. So everyone, I'm sure, is using a differential extraction method in their laboratory. It's pretty much the gold standard. And actually, Pat will go a little more detail in differential extraction if you don't remember, so I won't go through it. But some of the other separation methods that are out there, flow cytometry, it does an okay job in separating, but unfortunately you need a lot of sample using flow cytometry. There's also a lot of new research in microchip separation, and even there's many funded NIJ research grants right now on that. And it'll be kind of interesting. I'm kind of excited to see where that is going. Membrane filtration is another kind of simple method to separate sperm from epithelial cells. Again, so far you kind of need a lot of sample in order to do that. And then there was also work on magnetic antibodies, trying to use like magnetic beads attached to antibodies to separate your sperm cells. The problem with that tends to be that the persistence of the antigens on the cell surface of the sperm, especially in forensic samples. And I just added this because although Y chromosome analysis isn't really a separation method, it is obviously used when you have mixtures. So, but it's very different. Statistically, of course, you won't get as good of a power of discrimination as you would if you were able to do your STR loci, your 13-core loci. So we propose, of course, that laser microdissection is an alternative method for separation of sperm from epithelial cells. And I apologize, this is kind of an, oh, what did I just do? I bumped that. There we go. This is, I accidentally got an old PowerPoint slide in here, so I'm going to draw some things in. This is a kind of a basic diagram of what the LMD instrument, at least the Leica instrument, is. Here is the microscope objective, and you have the laser coming through the objective. And down here is you have your slide, and this is an example of the slides you have. But here I haven't drawn. There's actually another layer in here. The pen film. So you have that slide, and if you look at it, you can see that it's actually, that membrane is glued along the edges, and then in the middle it's not actually glued, it's just kind of suspended there, held there. And you prepare your specimen on that slide, and then you stain it, or however you decide to prepare it. We'll talk about staining later. And in this diagram, there's actually an example of just a couple little dots. Let's just say that those are the cells. And underneath the stage is a tube, and you'll notice that the slide is upside down. So the laser will come through, cut around the sample, and then the sample will drop with the membrane in it into the tube. That's how you collect your sample. Okay, so here's that epithelial sperm cell mixture again, and this is my kind of homemade version, not the fancy Leica version. Oh, actually, no, there's a couple things to talk about. So what are the technical issues of sperm ID using LMD under the bright field conditions? And I talked briefly about this right at the beginning, that pen film architecture, the pores that you will see. So a stain is necessary to really, some of the tests that I did earlier, I really did try to do it just unstained and cut out sperm from epithelial cells. And I did have somewhat of an error rate, per se, that I would get a mixture, because apparently I wasn't able to see maybe all the epithelial cell nuclei and avoid them. So I think a stain is necessary. It also helps to adjust the focal plane, like Kelly said. And there are also some new slides that were just available this spring, and we're actually going to kind of test those out that do not have this architecture. What about high density of epithelial cells? What do you do when there's just a dent, and how are you going to find those sperm and cut them out? There's some different techniques we've talked about diluting out your smear and spreading it over a large area. When we're in the lab, I'll talk about maybe possibly ablating some epithelial cell nuclei prior to collecting sperm that are near these cells. And also perhaps even a little mini lysis step that you do before you spot your pellet. So again, dilute pellet over a large area is an easy method. And then there's no cover slips. So these are just kind of some obstacles that will be a little different than what you might be used to. Remember, you're looking through actually the glass, and just kind of one of those little things that sometimes people forget. The correction collar, there is a correction collar on these microscopes, so you can adjust, and you have to adjust for the 1 millimeter of glass. So this is a picture of what you'll see on the computer screen. This is an older version of the software, what Kelly and I and Pat have. You'll have a whole bunch of controls available to you. And in this picture, the circles represent, the analyst has actually designated which sperm they're going to collect. And after dissection, this is what it would look afterwards, cut out. And on the right is looking into the tube cap where you've collected your sample. So these are actually little teeny sperm head. You can also do epithelial cells just as easily. And if you notice, this particular cell is actually this one that was in the tube. It's kind of the same shape, but it's flipped. The current software, this is, I just put this in so you have that information, is very similar, has some more features which are great. We're very excited about these new instruments. Kelly, Pat, and I have seen how much, you're lucky, how much faster that they are than our older instruments. And some nice features that maybe little technical difficulties that we may have had in the past are solved with the new instrument. So again, this is I talked about this before. This is my homemade video. This is what it'll look. You'll see that the pen slide down here. And you see those little circles? Those are actually just circles I put on there so I know where my sample is sitting. I use a simple office Avery labels, the kind you use for reinforcement of your 3-hole punches. And that actually makes a 7 millimeter spot, which a lot of people like kind of that working volume, and I put 3 microliters in each of those. In addition, those little labels are helpful when you're staining to kind of put your drop in there, and it kind of holds that so you can easily stain that. Let's see if I got this. I'm gonna have to, let's go back. Oh, you know, that's right. I have the, let me close this out. I have the video actually on a separate file. Let's see. Nope, that's the other one. This one. Oops, sorry. Don't know how to maximize that. Okay. So right now you'll see this kind of light going on. Do you see that little blue mark in there? That's actually the laser. It's hitting the sample. It's cutting. And so what's happening is it's going through the slide, cutting around, where the membrane is, and the membrane and the sample are dropping into the tube. And so this is a kind of poor quality video. I apologize. This is kind of the speed at least the old instrument was working at. Analyst is identifying the sperm cells and activating the laser. Then the laser is cutting around the sample, and the membrane will be falling down into the tube. And at the time, I was timing how fast can I cut cells, and in about 20 minutes, I could cut about 100 cells if it wasn't a difficult sample, just kind of an average sample. Kelly, I think, was timing some people the other day, and it was much faster. And then you can also collect epithelial cells, obviously in a different tube. You'll notice actually the color of the pen is different. And you can collect an entire cell body or just the nuclei, as demonstrated there.
Are you directing the path of the laser manually, or is it automated to follow the line you've drawn?
It's automated to follow the line. Now the features are you can do that line draw, and then you hit a button that says cut, and then it automatically cuts these things, or the circles. You also have rectangles or squares, but I have never used actually that feature of the instrument. But now you also have a basically move and cut where you could just hold your mouse down and use the laser manually. And you'll also see a new pen mouse. You can actually go around in the screen and cut manually. Those are nice features to have. Okay. So any questions on that? That's just kind of before you go into the lab, just to kind of get a feeling of what's going to go on.
Actually, I have a quick question.
Yeah.
Do you have any information on long-term storage of those slides?
We were having this discussion yesterday about what kind of validation we may have to do with long-term storage and what might be necessary. Well, we should talk about. We will talk about in the round table. We were thinking actually we do not have any studies right now, but it's something that we're thinking about how are we going to design this.
I was going to say, I think in the novel validation guidelines it talks about stability, testing stability, and so of course I'm saying, well, is that going to be testing the age of the slide, the age of the sample on the slide, right. And I would think depending on your laboratory, of course, I'm not sure if these are all, if other laboratories validate, are those going to be considered internal validations, or are they going to be novel if someone else published? Hopefully we'll write a paper about what we've done. But yeah, but I was thinking, at least for our laboratory, we keep what we call our pre-amplification work product for 6 months, and we put this in our report before it goes out, and so therefore any agency or whatever can come and get it if they want, but then after 6 months we destroy it. So I said, well, we probably need to at least test these slides after 6 months to see what's happening to the sample. Is it still good? Maybe I'll do a year, because I know I have some slides that are a year old, so I can probably still go back and test those. But yeah, I'm thinking we probably need to do some just age of that sample.
We also have a policy in place about saving sperm-positive slides. So if we don't cut them all out, I'm just thinking in terms of what are we going to do with the rest of the slide, and if that film kind of comes off over time, or if it has to be stored at a particular temperature to keep it intact. So.
And that's funny. That's a difference between Christine and I. I was telling her I've stored all my slides just at room temperature in a box. Mm-hmm. Okay. Now I'm not doing casework slides yet, but just slides in one of those big boxes, slide boxes at room temperature, and they seem to store fine.
And I store them frozen in these things, like kind of each little project I put in these. So
and then she dries them out. She dries them out. Yeah. But there is an issue with water. If the slides aren't completely dry, water can kind of bubble up that film underneath. But yeah, so those are things that still have to be addressed.
So I just wanted to briefly go through what we're going to be doing in the lab, and you might want to make some corrections. You have this sheet in your book. It's probably toward the front more than anything else, not before all the PowerPoints and the articles. Yes, lab practical flowchart.
It's right after the LMD self-selection worksheet. right after that. It's near the front.
So you see, there's a lot of behind-the-scenes work going on, and Carrie isn't here, but Carrie has been working in that lab. Late at night, too. Before you got here, she was preparing these mixtures for the laboratory. And so you just know what kind of mixtures are you dealing with. Kind of made them a little easy for you guys. We were mixing one buccal swab with about 5 microliters of neat semen. And she was obviously eluting the cells off of those and preparing the cell pellets. Now, you students will not be preparing these first nuclear fast red slides because the first class did that for you. You will be preparing some different slides with the director slides to kind of get a little experience with that a little later. But I just wanted to show you, does someone have one of those slides that I passed around? Yeah. So if you noticed on these slides, and I don't think you can even really, no, you can't. I don't know if you can see on these things. Actually. Can you notice that there's like a kind of a rectangle in the side? There's a film covering the entire slide, but it's glued or adhered to on the edges. And you want your smear in the rectangle. And what like I said, remember I told you I had these Avery labels. You can actually stick those on, and what you would of course do this all with gloves because these samples, remember, are going to go for DNA analysis. You want to stick those on there. Usually, hopefully, you can get 3 on there. Wow. It's kinda hopefully, you can get 3 on there. You can actually if you get them all in there. And that's what I like to put 3 circles on there and do 3 microliter spots. And my cell pellets are usually 100 microliters because I want it a little diluter. Probably many of you are probably below 50, 30 is your cell pellet. My recommendation is make it a little more dilute. Remember I told you that what if you have a high density of epithelial cells? And you have 3 spots to work with too. And if you found that you didn't even have enough there, you could make another slide. But so far, I haven't had to go to make a second slide. One thing, couple of points to when you're preparing these slides. This membrane can be somewhat fragile, and when you put these labels on, you don't want, the minute you place it, you don't want to be like, oh, I need to replace it, because you don't want to tug at the membrane or poke the membrane, because if you poke it, you can get water in there when you're doing your staining, or any kind of liquid for that matter, and then it will be really hard to have to dry out that slide, and it's going to affect your cutting when you have this water floating around. That also goes for when you actually spot your sample on there. You don't want to just put your pipette tip right on that membrane and just put the spot. Be very gentle. So then we're actually going to be going into the lab shortly, and Andy Lee's first going to give kind of what he called care and feeding of the instrument. Very short, just the basics of the instrument, and not to make it explode or anything like that, but don't worry. And then Kelly and I will break you up in smaller groups and really go in more detail how to cut samples out. Then you will actually be collecting sperm samples, and we'll break you up in smaller groups to do that. You'll actually be switching off. You'll see that you'll be performing LMD on some of the mixture slides. And there's this also fluorescent examination. This will be going on at the same time. When half of you are doing dissection, the other half will be preparing the fluorescent slides. Then you will switch off. Then Carrie at NFSTC here, she will be completing the cell lysis, basically be handing off the samples. She'll be completing the cell lysis, performing the PCR, and be aware, okay, we're doing 50 microliter reactions, but we're doing 34 cycles. Okay, we don't have this really lab set up totally for low copy number work, but just try for just exercise purposes, try to be as careful as you can. At least you must wear gloves in all of the process. There are masks in there if you feel like that you're some kind of shedder, and also when you're doing any kind of your tube loading and pulling samples in and out, it's a good idea not to be talking. And then overnight, or tomorrow morning actually, Carrie will be running the samples on the CE, And we will be back in the lab in the meantime going over some fluorescent examination of the slides that you prepared the day before and some additional slides that were brought. We'll also look at some advanced operations and some new software that for auto recognition. And then hopefully the data will look good, and later in that day we will we'll look at the DNA data. So we are ready to go over to the lab, and Andy will give you a demonstration